I'm Bello Danmallam, and I'm delighted to present on the topic ecological impacts of urbanization, insights from the African Bird Atlas project in Nigeria and Kenya. So this presentation is part of our collaborative effort with the Tropical Biology Association, Kenya Bird Map, and the Nigerian Bird Atlas project. All right, so urban areas are growing across Africa at an alarming rate. And by 2030, it is projected that these urban areas will triple in size, accelerating human-driven environmental changes and threatening bird life, as evidenced by research. So already over 50% of the global population lives in cities, and by 2050, it is expected that this number will grow to 70%. So, and this could have a very high impact on birds and biodiversity. So Nigeria, as a country, is an epicenter of this global change, as we can see on the map with the human footprint index, which shows the extent of human impact on the environment, and Kenya with a moderate human impact on the environment. So what? Why does it matter? Why should we care? As we know, birds play crucial roles in the environment, roles such as the pollination services that they play as scavengers, helping to remove carcass from the environment, seed dispersal by hornbills, and pest controls by birds like insectivores, as well as raptors, helping to control rodents in farmlands and in the environment. So increased human activities such as urbanization and agricultural expansion will actually disrupt these vital ecological functions that these birds play. And the loss of these birds in the environment could mean reduced pollination services, reduce dispersal services by hornbills and the rest, as well as accumulation of carcasses in the environment. So we use the African Bird Atlas project data specifically for Nigeria and Kenya. So the Nigerian Bird Atlas project was established in 2015, and the Kenya Bird Map was established a little bit earlier in 2013. So these two projects are part of the African Bird Atlas project. And observers, we have citizen scientists all across. So these citizen scientists help by collecting data within a pentad. A pentad is a 5-minute by 5-minute square, as the name implies, a pentad, which is approximately 9 kilometer by 9 kilometer grid. So observers spend a minimum of 2 hours to observe birds within the pentad across habitats within the pentad. So observation of up to that 2 hours is known as full protocol observation. So we use full protocol submissions, full protocol cards, at least 4 full protocol cards submitted to this database for Kenya and Nigeria. So we extracted the data of 914 species in about in 227,420 records for Nigeria, and 1,069 species in about 265,276 records for Kenya. Very large data. So these two countries lies in two strategic locations. The East Atlantic Flyway, where Nigeria lies, and East Africa, East Asia Flyway, where Kenya falls. So this ensures that we capture migratory birds in our analysis. So we populated our bird data with the functional traits as obtained from Child et al. And Lee et al. 2021, respectively. So roles such as scavengers, grazers, scavengers helping to helping in carcass removal, disease control, and the rest, grazers, raptors, amongst others. So we populated our data with these different roles that these birds play in order to be able to calculate functional diversity. So these are the metrics that we computed, taxonomic and functional metrics. Taxonomic richness is the total number of distinct species within the community. Functional richness is the volume of functional traits, is the range of functional traits within the community. And we have Rao's quadratic entropy, which is the overall functional diversity that combines the trait differences, the differences in the traits of these birds and their abundance. So this particular metric, the Rao's quadratic entropy, factors abundance. So our abundance in this analysis is the reporting rate. So we use the reporting rate as a proxy for abundance. Reporting rate is the number of times a species is observed per pentad per our observation unit. So the taxonomic diversity is simply Shannon-Wenner diversity index. So 4 diversity metrics, taxonomic diversity, functional diversity, taxonomic richness, and functional richness. So we use the human modification, human footprint index, which was obtained from NASA's socioeconomic data. So this human footprint index provides a cumulative measure of the human modification of terrestrial lands. It's a continuous variable between 0 to 1. The higher the value, the greater the human impact. This metric combines about 13 anthropogenic stressors, including population density, built-up areas. We have croplands and the rest to arrive at a metric of 0 to 1. So we use this as a human impact index. So we use Bayesian models. So these Bayesian models can incorporate prior knowledge and structure into the analysis. They are able to account for expert knowledge. So we use the default priors within the BRMS package in R. These default priors are weakly informative, allowing our data to drive the inference. So these Bayesian models, they are not limited to p-values. So instead, they provide probability distribution of parameters. So we ran 2,000 iterations each, 4 chains, 2,000 iterations, and the first 1,000 iterations were used as warm-up. So you can think of it as running a computer simulation 4 times, and each with about 2,000 steps or repeated process. So 4 chains with 2,000 iterations. So the first 1,000 steps in each run were just for practice. That's what it meant by warm-up. So the first 1,000 steps were just for practice to help the simulation settle in a very good starting point before collecting real results for our analysis, for our results. So we answered this question: how does human modification and urbanization affect avian taxonomic and functional diversity in Afrotropical birds? So this is the causal structure showing the interplay of the different metrics that we use: the human modification index, 0 to 1, and human settlement in 4 categories: the mostly uninhibited areas. We have the rural areas, suburban areas, and then city areas, a gradient of urbanization. And we have this data, probability of urban expansion up to 2030, which was also obtained from NASA's Socioeconomic Data and Application Center. So this shows the interplay, how these variables interact to affect taxonomic and functional richness and diversity of birds. As we know, human modification to build areas, to build up human settlement, there has to be human modification of the environment. Likewise, with more built up areas, the probability of urban expansion increases, which affects the taxonomic and functional richness of bird communities. So we found a total of 900 species across 89 families in Nigeria, while 1,069 bird species in 94 families were recorded in Kenya. So out of this, 90% were least concern in the IUCN Red List, 27 species threatened with extinction in Nigeria, 3 critical endangered, 11 endangered, 13 vulnerable, and we recorded 44 threatened species in Kenya with 5 critically endangered, 20 endangered, and 19 vulnerable species. So in all, we recorded 179 species as habitat specialists in Nigeria, 225 habitat specialists in Kenya, 40 urban adapters in Nigeria, and 50 in Kenya. So this shows the effect of the settlement types, the gradient of urbanization on the bird communities. So from figure A, we can see that taxonomic richness, and figure B, functional richness, decreased along the gradient from rural areas to suburban and city areas, indicating fewer species and threats in more urbanized environment. So as we can see, mostly uninhibited areas, the first bar there had showed a slightly lower richness, which is likely due to lack of habitat heterogeneity in these mostly uninhibited areas or some level of human activity. So, however, we could see that for functional diversity, there is a gradual increase from uninhibited to rural to suburban and to city areas. And this could be as a result of the filtering effect of urbanization, where generalist species are being favored as compared to habitat specialists. So these generalist species like the doves and pigeons can proliferate in numbers, which has an effect. As I mentioned earlier, this Rao's quadratic entropy, the functional diversity metric, can easily be inflated by the abundance of certain group of species. So in this case, the generalist species that are mostly adaptable to urban areas will inflate this metric, influencing the results. So using the same data from a separate study with matched pentats in protected and unprotected areas, we found high variability in human impact within protected areas, suggesting that protected areas are not entirely free from human influence. As urbanization displaces birds, as we've seen from urban areas, they move into other areas. Yet, even within those areas, including protected areas, they may still face threats such as habitat degradation or even poaching. So, so as we can see here, with the high variability in human footprint within the protected area, although the unprotected areas had higher human footprint when compared to protected areas, but the protected areas are not safe with the high variability of human footprint. in protected areas in Nigeria. So this shows the posterior contrast between the peers, the different urbanization categories. So here you could see that the C and D are the relationship that stood out, the differences that stood out. They have been marked in light blue. So we found that rural areas had higher taxonomic diversity as compared to cities, significantly higher. Likewise, suburban areas had significantly higher taxonomic diversity as compared to city areas, and uninhibited areas. Here, we can see uninhibited areas having higher taxonomic diversity as compared to city, with the positive difference. So positive difference here implies that the first term, the first parameter is higher in value compared to the second parameter. In this case, as we've seen, rural area, the first parameter had higher taxonomic diversity as compared to city, same with suburban, city, and uninhibited as compared to city. However, when it comes to functional diversity, there is a flip where there is a negative difference. So meaning that city areas had higher functional diversity, the Rouse quadratic entropy value. City areas also had higher functional diversity as compared to suburban. Likewise, rural areas had higher values as compared to uninhibited areas. So, and this could also be as a result of biotic homogenization effect of urbanization, where specialists are being driven out of urban areas and being replaced by generalists in high numbers. So inflating the functional diversity metrics within these areas. So we also found out that human modification had a predominantly negative relationship on taxonomic richness, functional richness, and taxonomic diversity. As you can see, as human modification increases, there is a gradual, a steady decline in taxonomic richness as well as functional richness, the number of different roles birds play in the environment. Likewise, with the taxonomic diversity, there is a steady decline taxonomic diversity with increase in human modification. However, when it comes to functional diversity, there is also an increase. There is an increase, rather. So this can also be attributed to the filtering effect of urbanization, where generalists are being favored, inflating the functional diversity metric in our study. So here is the relationship between the probability of urban expansion and taxonomic richness, functional richness, taxonomic diversity, and the functional diversity. So there is a complex relationship here, as initially there is a gradual decline, and then there is a rise in taxonomic richness function. You could see a pattern here. Taxonomic richness, functional richness, and taxonomic diversity follow the same pattern, similar pattern. So there is an initial decline followed by a rise, but at 75% probability of urban expansion, there is a rapid decline in these diversity metrics, taxonomic richness, functional richness, and taxonomic diversity. However, when it comes to functional diversity, there is a gradual increase in functional diversity. So, and this could be as a result of the adaptive potential of some species, group of species like the dove and pigeon, inflating this metric. Urbanization significantly reshapes natural habitats, as we've seen with the human impact, the human modification index, and this reduces the availability of suitable areas, and in the process driving species out of urban areas. And we also saw that the protected areas had a very high variability of human footprint, meaning that they are not safe as well for these birds. So where the question is, where do these birds go? Where do our birds go? So here is a case study of the Ogunpa Forest Reserve in Nigeria. So the Ogunpa Forest is one of the oldest and last standing forest reserve in Ibadan. Ibadan is one of the cities in Nigeria, in Oyo State. And this particular Ogunpa forest played a very crucial role in biodiversity conservation. It helped in flood control as it lies within a watershed, a floodplain, and it also helped in urban cooling. So, however, the current deforestation in this Ogunpa Forest Reserve, as depicted here on the slide, this is currently ongoing here in Nigeria, in Oyo State, could threaten the biodiversity within this lush habitat, within this lush forest reserve. And this particular area is being cleared for residential estate development by the Oyo State government of Nigeria. So even though the environmental groups here in the country, the different conservation organizations have argued that there was no proper environmental impact assessment that to clear up this place. They've written a number of letters to the governor, and still this havoc is still ongoing. So this highlights the environmental injustice, as we know that local communities are the ones to bear the consequences of this ecosystem loss. Yeah. So advocacy efforts are already in place, and we just hope that things will change. So this just highlights the complex, the struggle between urban expansion and habitat preservation in developing areas like Nigeria. So in a nutshell, balancing urban development with habitat preservation is very essential to prevent further biodiversity loss in our cities and to maintain ecological functionality in urban landscapes. So, and this can be done by keeping green spaces and natural areas in these urban areas, in cities. And then we recommended that park managers should strengthen their protection efforts with stronger buffers and corridors to implement effective management practice and enforce regulation to sustain high taxonomic and functional richness of birds. So in summary, we found taxonomic richness significantly affected by urbanization. It decreased. And likewise with functional richness, there was a decline in functional richness with urbanization gradient, as well as taxonomic diversity. So this shows the predominant negative impact of urbanization on bird communities. However, with functional diversity, we saw an increase in functional diversity as urbanization increases. So there is an urgent need of conservation measures to fulfill the target 3 and 4 of the biodiversity, the Kunmin-Montreal Global Biodiversity Framework of protecting 30% of land and waters by 2030. That's the 30 target, and halting species extinction. So, and to do this, collaborative action is essential, involving species specialists, conservationists, and everyone. Conservation is everyone's business. So all hands must be on deck to save birds and biodiversity. So this paper is out. You can get it online, open access in Frontiers in Conservation Science. You can easily download it. So, all right. Thank you very much for listening.